Hello and good morning. Today our topic is the physiology of kidney, and these is the following points which we'll be discussing today. First of all, the first point is the filtration of blood. So the glomerulus is the site of filtration of blood. This is the glomerulus. Then after it comes the proximal convoluted tubule, and then the loop of Henle, and then the distal convoluted tubule, and then the collecting duct. Okay, so the Bowman's capsule consists of the basement membrane and the podocytes. Okay, it filters blood. So let me show you first what is a podocyte. As you can see, this is a podocyte, and this is a cell body, and these are its pedicels connecting each other. Podocyte fold processes. Okay, and this is the basement membrane, and it has slit-like pores from which the blood cannot pass and proteins cannot pass. Only small molecules like glucose etc. can pass, urea etc. Okay, and what it does is, the Bowman capsule does is, it filters the blood and it holds back large molecules such as proteins and blood and red blood cells, and passes through small molecules such as water, salts and sugar, and it aids in the formation of urine. Okay, podocytes, which are known as visceral epithelial cells in the Bowman's capsule in the kidney that can wrap around the capillaries of the glomerulus, leaving slits between them. Okay, these are little slits between them. And they are also involved in the regulation of glomerular filtration rate, GFR. When podocytes contract, they cause closure of filtration slits. This decreases the GFR by reducing the surface area available for filtration. Basically, when the podocytes contract, there is narrowing of the filtration slits, and the less amount of things pass through it, and then there's the GFR is low. So maintenance of renal blood pressure. This process is achieved by the juxtaglomerular apparatus. Okay, what is juxtaglomerular apparatus? It consists of juxtaglomerular cells, macula densa, and extraglomerular mesangial cells. So this is just a juxtaglomerular apparatus. As you can see, this is the Bowman's capsule, and this part, it consists of macula densa cells in the distal convoluted tubule. Okay, and these cells are called the granular juxtaglomerular cells. Okay, function is the release of renin, and the stimulus is low blood pressure. When there is low blood pressure, there is the release of renin. Then it converts the angiotensin into angiotensin 1. Then angiotensin 1 is converted to angiotensin 2 by the angiotensin converting enzyme ACE. And functions of angiotensin 2 are, it acts as a vasoconstrictor to raise the blood pressure. It stimulates the release of aldosterone hormone from the adrenal cortex. What aldosterone does is, it stimulates the distal convoluted tubule to reabsorb salt. If NaCl is reabsorbed, water will follow. H2O will follow. So when H2O will follow the salt, there will be the retention of water in the body, and it will alleviate the blood pressure. Okay, so the reabsorption of salt, how it happens? Basically, I told you this before. It induces the movement of water to the blood by osmosis, thereby raising the blood volume and hence increasing the blood pressure. So what is the function of macula densa cells? It monitors, basically, if it tells basically the blood pressure is low or high. If the concentration of the salt is raised, the macula densa cells inhibit the release of renin, and there is no angiotensin 2. Okay, so no renin, no angiotensin 2, no aldosterone, blood pressure decreases until it is sent back to normal. What is the function of extraglomerular mesangial cells? The third part, it secretes erythropoietin. Okay, what basically erythropoietin does is, it is a glycoprotein hormone which controls erythropoiesis or red blood cell production. Basically, erythro means red and poiesis means production. Okay. So this is basically the whole chart. Blood pressure is falling and angiotensin 1 and then 2, and this is the enzyme which converts 1 to 2, and basically renin converts angiotensin to angiotensin 1. Okay, then it causes two stimulus, rising of the blood pressure and functioning of stimulating the aldosterone. Okay, and then there's the salt retention. How urine is formed? There's ultrafiltration, reabsorption, and secretion from the blood tubule. Okay. What is countercurrent multiplier? As you can see, when we go down and then we go up, this is the basically descending limb and this is the ascending limb. Descending limb is passively permeable to water. What we extract water and its osmolarity is increased, and when it reaches the descending limb down there, it is maximum, and then when we are going up, we are losing NaCl. Basically we are pumping out the active transport of sodium, and the chloride follows it passively, and then because of that our osmolarity milliosmoles falls. Okay, and we finally go to 100. Okay, this is also the explanation of countercurrent multiplier, which I told you. Basically see, Na is going inside, it is being actively pumped out of the ascending limb into the interstitial fluid. Okay, basically this is the interstitial space. Okay, and this is Na being pumped out, and when Na goes out, Cl follows passively. Okay, when this is the Cl following it passively here as well, going out with Na. Okay, and it increases the NaCl concentration by 4 times outside, and water seeps out of the descending limb into the interstitial fluid by osmosis. Okay, okay. How regulation of electrolytes, how it is done? Electrolytes are basically the substances that becomes ions in the solution and acquire the capacity to conduct electricity. Basically, there are ions of electrolytes are sodium, potassium, calcium, magnesium, and chloride, and hydrogen phosphate and hydrogen carbonate. So maintenance is occurred, and the osmolarity of both the fluids and the urine is the same. So what is antidiuretic hormone? It is also known as vasopressin. It is released by hypothalamus, and stimulus is low blood level. It prevents the production of dilute urine, helps in the reabsorption of water in the kidneys, and maintains the osmolarity of plasma levels normal. Okay, what is acid-base balance? It helps in the human homeostasis. Okay, it is important to maintain cellular stability. And in acid-base balance, the kidneys is responsible for major 2 activities: reabsorption of filtered bicarbonate. If body is getting low bicarbonate, it reabsorbs it, and it excretes the fixed acid like acid anion in associated hydrogens. Okay, and this is the cells in the walls of tubules. These are the walls of tubules, and this is the developing urine, and this is the blood. Okay, so hydrogen is passing into the developing urine, and sodium is coming inside because we need a retention of water. Okay, and SCO3 is also coming inside because we need to maintain the homeostasis in the body, the acid base balance. Okay, so formation of hydrogen, its secretion is from the organic acid and base secretory system, uric acid, antibiotics, and diuretics. Okay, so basically the bicarbonate is converting it to hydrogen and SCO3. So in case of high hydrogen, it is bound to ammonia, phosphate, and bicarbonate to form NH4 and this. What is reabsorption of materials? Nutrients such as glucose, amino acids, and other metabolites are reabsorbed because we cannot lose it. We cannot lose them. Glucose is our precious source. We cannot lose it. Okay. They are absorbed in the medulla, and they are very important. And its location is the convoluted proximal tubule situated in the cortex of kidney. Basically, it is absorbed first most so that it does not get lost in the later parts. Okay, because it's very precious. The glucose is very precious to us. And excretion of substances, how it is done. Calcitriol, activated form of vitamin D, promotes intestinal absorption of calcium and the renal reabsorption of phosphate. Okay, so basically calcitriol helps us in this. Okay, and renin helps us, and erythropoietin helps us as well. ADH helps us in water vasopressin, it is also called. The prostaglandins, kinins, and 1,2,5-T3. So how the wastes are excreted? First of all, we excrete these things: urea, uric acid, creatinine, and products of HP metabolism. Okay, and metabolites of hormones, foreign substances such as drugs, pesticides, and other chemicals ingested in food. Okay. Plasma clearance rate. What is plasma? Plasma is basically the blood. And what is clearance rate? It is defined as the amount of blood cleaned of a substance per unit time. Different blood substances have different clearance rate. Okay, it is the function of glomerular filtration, excretion of the peritubular capillaries to the nephron, and the reabsorption from the nephron back to the peritubular capillaries. So how to find the plasma clearance rate? C is equal to V multiplied by U divided by P. What is C? C is the plasma clearance rate, C for clearance, V for urine production rate, U for the construction of a substance in urine concentration, and P for the concentration of substances plasma. Okay, U for urine, P for plasma. Okay, and V is for urine production rate, C is for clearance, and its rate is ml per minute. Okay, we will do pathology in a separate lecture. Thank you for listening, and please like, share, and subscribe.